So this is what genetic screening is. It's a population-based method to identify persons with increased susceptibility for a genetic disease, regardless of family history or independent of clinical status. So genetic screening is based on individual genetic tests, and we talked about genetic tests for this and that, and we're going to talk more about those today. The difference between genetic screening and genetic testing is sometimes these two terms are used interchangeably, but if a difference is made, genetic screening refers to a population level screening, while genetic test is an individual test applied to a single person or to a family. So like I said, genetic screening is based on genetic tests, and these genetic tests have to meet some criteria to be effective, to make genetic screening effective. So this is the list of criteria for an effective test. So genetic screening is typically something that we do en masse. So it can be supported by local governments, or it can be mandatory, which means that most, if not all, newborns are subject to screening. But again, the screening uses specific methods, individual tests. And again, this is the list of criteria that such a test needs to meet. So first, there has to be analytic validity. So there has to be a test, a rapid and relatively inexpensive laboratory test available that detects the appropriate metabolite. So you will see that genetic screening in most cases is based on profiling of metabolites from blood. So we get a single sample, and from a single sample we retrieve information. We retrieve a profile of metabolites, and based on the level of each metabolite, we can establish if this is normal level or this is abnormal, concluding that a genetic disease underlies this abnormal level. There has to be clinical validity or justification. So the laboratory test needs to be highly sensitive, which means that no false negatives are allowed, and reasonably specific, which means that we expect few false positives. In other words, and it has some strict definition that we're going to go over. Such a test needs to have high positive value, a positive predictive value. So you may have noticed that when we talk about a sensitive test, we want no false negatives, but we can allow for a few false positives. And the reason is that false negatives are very costly in medicine. So if we overlook a true disease, then it has very serious consequences, right? Because a person, or let's say this is a newborn, that is a false negative, is genetically diseased, but it's classified as healthy and as such won't be treated, which again may lead to very serious consequences, including death, right? False positive, when we say that, that when a person is healthy but we classify it as sick or a carrier, let's say, then it's also serious, but the cost is not as high as in the case of false negatives, because it may lead, so what's the cost here? It's typically a distress when a family finds out, let's say, the baby has a genetic disease, but it turns out at the end that it does not, because this is a false positive. Of course, like I said, there's some distress involved, but the cost overall is lower compared with false negatives. Does that make sense? This is a digression. I want you to understand really what sensitivity and specificity of a test, of a genetic test in particular, means. This is a simple 2x2 table that summarizes results from a hypothetical test. So let's assume that there is a test for obesity based on blood work. Okay, so we test blood, we look at some metabolites, and based on the molecular level of these metabolites, we either say that the person is overweight or is not. So we have the test result, yes and no, overweight and not overweight, and then we have the true condition that we can check, right? So let's say that this is a test to see how effective this blood test is, and we know the true condition, overweight and not overweight. So there are 4 possibilities, right, with these 2 variables. So the test can be positive, and the person can be truly overweight, which is true positive. And this combination of these 2 variables is what we call sensitivity. Then the test can tell us that yes, the person is overweight, but the true condition is not overweight, which makes such a case false positive. Those of you who took statistics may remember that this type of error is called type 1 error. And then we look at negative results of the test. So overweight, the answer is no from the test, but the person is overweight, which makes it false negative or type 2 error. And finally, negative test and not overweight, which makes it true negative. And these true negatives are what we call test specificity. So in other words, sensitivity quantifies the avoiding of false negatives, and specificity does the same for false positives. So specificity is the avoidance of false positives. So ideally we want a test that has high sensitivity and high specificity. But typically there's a trade-off between these two. There's a trade-off between sensitivity and specificity. Trade-off means that there's like a negative correlation. So typically in real life what we have is that high sensitivity is associated with low specificity, which means that we allow for the cost of some false positives, but we want to avoid a high sensitivity. We want to avoid false negatives because the cost of false negatives is high. So just to give you, so I mentioned medicine and like typical approaches to genetic tests in application to medicine, but you can think of we can think of other real life examples, like let's say scanners at airports. They are set to trigger alarms on low risk items like keys or belt buckles, and we can say that this is that we have many false positives here when a scanner like that triggers alarm on small items that are not creating really risk. But the reason is that we don't want to miss true items that pose risk, right? So that's why the test is highly sensitive at the cost of low specificity. So it applies to scanning systems for security reasons, but of course it also applies to medical practice. Does it make sense? So let's get back to these criteria for an effective test. So that was a digression. What a sensitive and specific test means. So you need to have a good understanding of these terms. So now in application to like massive testing, which we call genetic screening at the population level, there must be some clinical utility, including treatment. So it makes sense to pay for population wide tests if treatment is available. Otherwise, what's the point? We just increase distress in the family. We cannot really provide any treatment. So it's not. It doesn't make sense to invest in a test like that. The next one is that typically we want early institution of the treatment when it's available, before symptoms become manifest, which reduces or prevents a severe illness. The next one is obvious that routine observation and physical examination will not reveal the disorder in the newborn. So that's why a test, like a metabolic test, is required. And finally, the condition is frequent enough and severe enough to justify the expense of screening. If something is extremely rare, then of course it's not justified. It's not cost effective. So this is a short list of the best known population screening efforts in genetics, and those are in many instances government supported or even government mandated programs to identify pre-symptomatic infants with diseases for which early treatment can prevent or at least reduce the consequences. The table is organized based on the frequency of these conditions. All of them except the first one, which is congenital hearing loss, are based on biochemical blood tests. The first one, the hearing loss, is of course based on direct hearing test. So this is not a molecular test. You want to remember those that are most common, and at the same time those that we discussed, or we already discussed, or are going to discuss today, like sickle cell disease, hypothyroidism, phenylketonuria, which I'll tell you more about today, and so on. This is a list of 28 diseases that are tested in Virginia. So this is the newborn screen, and some of those that were listed before, or actually most of them are included in this list, including phenylketonuria or PKU, sickle cell anemia is here, other hemoglobinopathies are also tested, including sickle beta thalassemia, sickle hemoglobin C disease, and so on. We also talked about cystic fibrosis. So you want to remember those that we discussed in more detail. Of course, I don't expect you to memorize the list. Just to give you a sense of what we're talking about and how prevalent these tests are, and like what numbers of conditions are under these tests. So for many years, this newborn screening was performed by specific tests for each individual condition. So we had to draw more blood, for example, and run all these tests. So for example, this PKU screening was based on a microbial or a chemical assay that tested for increased phenylalanine levels. And this situation has changed dramatically with the application of a high throughput technology known as tandem mass spectrometry, TMS, which, like I said, provides a profile of metabolites that are informative with respect to all these conditions that I listed in my previous slides. That includes phenylalanine, which is one of the amino acids, and its increased abnormal accumulation leads to this phenylketonuria, PKU. This is a prototype condition that satisfies all of these criteria that I mentioned before. And for decades, these elevated levels of phenylalanine were detected in a spot of blood on filter paper and was obtained right after a birth. And there's some microbial and chemical tests, quite tedious, that were taking place. And this, again, is replaced now with these high throughput mass spectrometry methods. So here you have the PKU pathway. This PKU, the major risk or presentation is intellectual disability, which can be avoided with treatment, or even relatively simple diet modification. This is described in the next slide. But before we go there, I'd like you to check this out. I'd like to emphasize the most important parts. So the main PKU presentation is due to the failure of phenylalanine hydroxylase, which is the enzyme that transforms phenylalanine into tyrosine. And tyrosine is required for multiple proteins and hormones, melanin, and so on. So when there's a mutation in this gene, phenylalanine level grows to abnormal levels. And this leads to PKU. There are some other less frequent varieties of PKU due to other enzymes that are involved in this pathway, like this dihydroapterin reductase and tetrahydrobiopterin. If they mutate, it also leads to phenylalanine increase, and some PKU variants that are actually more serious than the main one. And the reason is that we cannot really manipulate these forms. We cannot reduce these symptoms using diet changes, unlike in the main PKU, because these products, or these components from this pathway are required in the biosynthesis of neurotransmitters like serotonin, dopamine, and norepinephrine. But so it's very important to recognize which PKU form it is before we can apply treatment. And if this is the main one, like I said, it's relatively easy to control it, and that starts with diet. So high phenylalanine foods are avoided to control for PKU, and here we have a list of foods that are high protein and high phenylalanine. So this is something that is avoided. This is low phenylalanine diet, which is recommended for, or actually mandated for PKU, which includes vegetables, most fruits. Sugars are fine, and some low protein foods. So PKU is this iconic example of a genetic test that was incorporated into genetic screening, and you want to know exactly what it is, including some details with the genetic pathway, the molecular pathway, and how this can be controlled, including the diet. So that was genetic screening, which is different from heterozygote testing, and the differences are listed here. Heterozygote testing is applied when we screen for genetic disease in newborns, or for genetic susceptibility in patients. So sometimes a patient wants to know if she or he is a carrier, just to have a sense of the risk involved in having diseased children. So heterozygote testing is applied when there is relatively high frequency of carriers. at least in a specific population. And the test, of course, has to be available and relatively inexpensive. And we talked about that. It requires very low false negatives, ideally zero, and relatively low rate of false positives, even though the cost involved in finding false positives is lower than in the case of false negatives. There has to be access to genetic counseling for couples identified as heterozygotes, because then the risk is that they will have children being recessive homozygotes and affected. A prenatal diagnosis needs to be available, and of course there has to be some acceptance for voluntary participation by the population targeted for screening. And here are some examples of heterozygote testing, and you see that that refers to some specific populations in which certain disorders are increased in frequency. Ashkenazi Jews are one, and the next slide also refers to this population because it's relatively well studied. And Tay-Sachs disease, cystic fibrosis, familial dysautonomia, and Kenevan disease are among those that are most common in terms of carrier frequency. There is a form of Tay-Sachs that also occurs among French Canadians, and thalassemias are tested in Asians and the Mediterranean populations. Sickle cell anemia is more common among Africans. Cystic fibrosis is more common among Northern Europeans, and so on. You have now the column with test tells you what this test is based on. It can be based on biochemistry, which is about enzymes. There can be some enzymatic test, or it can be a test based on DNA, which is the direct information about mutations at the DNA level. Or there could be some blood work that detects the mutation, in which case this is called hematologic. So here's a panel of 19 conditions included in the current Ashkenazi Jewish carrier screening panel. I'd like you to pay attention to the most common, maybe the most common three, which is Gaucher disease, cystic fibrosis, and of course Tay-Sachs disease. Those are among those that are most common in the population. So some of these conditions were tested very early, like Tay-Sachs disease. Screening began in 1969, and the idea was to inform couples and recommend or not recommend marriages between young couples. And it sounds weird, and it was actually very controversial at the time, but it worked because this population or this community has arranged marriages, and there's the institution of matchmaker. So all these most of these marriages were arranged, and one of the main pieces of information used by a matchmaker was genetic test, where there are not persons who were seeking help of matchmaker were heterozygous for that. Like I said, that started with Tay-Sachs disease, so for Tay-Sachs, and it was criticized at the time. Like New York Times was comparing it to eugenics, that is kind of like discrimination based on genotype, which is something that is not popular. But for this particular population, actually it worked, because the frequency of Tay-Sachs disease and other conditions that are listed here actually dropped a lot, just based on arranged marriages. Here's another screening panel, just for cystic fibrosis. This is so-called ACMG screening panel. This acronym stands for the American College of Medical Genetics and Genomics. And like I said, it was constructed specifically to test cystic fibrosis. Cystic fibrosis is caused by mutations in CFTR gene, which is a huge gene with more than 1,000 mutations known that may cause cystic fibrosis. So this test was not perfect. The sensitivity was below 100%. Then there was an expanded panel that included, so the first test included 23 mutations. The expanded panel included 60 cystic fibrosis mutations. Combined, they had allele frequency higher than 0.1%. To give you a better sense of numbers involved in this ACMG screening panel, here's a table with distinction between ethnic groups, incidence of cystic fibrosis in these populations, and carrier probability without testing, just like before testing. This is carrier probability if negative for ACMG panel. So in other words, this column is false negative, right? So we ideally we want these numbers to be zero. We don't want any false negatives, but you can see that actually these values are not zeros. These numbers improve when this expanded panel of 60 markers is used, but still this test is not perfect, but it's still being used, because the alternative is to sequence the whole gene and analyze all individual sequences, which is very, very expensive, because this is basically genomics, expensive genomics or genome analysis. This test is more like an array. That there are some probes specific to mutations within this gene. So the test is relatively inexpensive and meets these criteria that we discussed before. Okay, so that leads me to genetic epidemiology, which is about how genotypes and environmental factors interact to increase or decrease susceptibility to disease. And there are three major approaches within genetic epidemiology: case control, cross-sectional, and cohort. Case controlled, and the definitions are here, is the least expensive and probably the most commonly used, is when individuals with and without the disease are selected, and the genotypes and environmental exposures of individuals in the two groups are determined and compared. The problem with case control is that it cannot be used to inform us about frequencies of this condition in the population because this is a non-random sample of individuals selected from a population. So because of this bias that we basically identify individuals that are potentially affected, introduces this bias to statistical analysis, preventing us again from getting information about the frequency at the population level. Cross-sectional is when a random sample of the population is selected and divided into those with and without the disease, and that genotypes and environment exposures are determined and compared. By environmental exposures, of course, it can be anything, including smoking, lifestyles, and so on. And the cohort is a sample of the population selected and observed for some time to ascertain who does or does not develop disease, and their genotypes and environmental exposures are determined and compared. The cohort may be selected at random or may be targeted to individuals who share a genotype or an environmental exposure. So this cohort gives us most robust information in terms of like statistical data. Also, the reason is that with cross-sectional is similar, but cross-sectional doesn't have this time dimension, which is important when there's, let's say, a very lethal mutation. That mutation occurs and it's followed by death shortly after. So this time effect is not included in cross-sectional, but it is in cohort. So like cross-sectional, because of this timing issue, may actually underestimate frequency of the disease in a population. Now there's some terms that I'd like to introduce, and I'd like you to remember. Now we talked about some of them, like sensitivity and specificity, and then positive predictive value. So let's define those rigorously, so you know what we're talking about. So let's start with a table for a disease association. So it's a relationship in a population between a susceptibility or protective genotype and a disease phenotype. So in other words, it's an association between genotype, which can be mutation and not mutation, or mutation wild-type, or like in here, it's susceptibility genotype present and susceptibility genotype absent. But again, it can be about this susceptibility, but in a broader sense, it's about mutation and wild-type. Okay, marker genotype, and then we have the condition, disease. It's individuals are either affected or they're not affected. So then these numbers are derived from a population. Those are counts of all these combinations. So we have 4 possible combinations for these 2 variables. One of them is genotype, the other is the condition. Okay, so I'd like you to spend a minute or so by checking this out because we're gonna go back and forth between this slide and the next slide, because these A, B, C, D stand for numbers and they're important. So A is positive genotype and affected individual, or the number of those in the population. So it's positive affected. B is positive unaffected. C is negative affected, and D is negative unaffected. Okay? And then we have sums of each, either row, which is A plus B for positive tests, C plus D for negative tests, A plus C for affected individuals, and B plus D for unaffected individuals. So I'll give you a minute to contemplate on that. Protective genotype is the opposite of susceptibility genotype. So sometimes we're looking for a genotype that actually increases resistance. But in a broader sense, in a more general sense, like I said, this is a marker for something, right? A genetic marker, and this variant, this marker is either present or absent. Okay, so we look at the same locus, at the same position in a gene. It could be like a single position, or it could be a genotype, like here. So we need some. You need to be aware of some flexibility here. That sometimes we talk about a point mutation, in which case would be the mutation is present. Here the mutation is absent. Sometimes we talk about a genotype, and like when this genotype is present, that makes the person susceptible to the disease, and this is the normal genotype, which we call susceptibility genotype absent, which means that the wild type, the normal kind of marker is or variant is present there. Okay, so this is the marker, and this is the non-marker variant, if you like. So let's start maybe with sensitivity and specificity, because this is something that I defined earlier, but now we can define it using numbers or these symbols. So sensitivity is, in other words, fraction of individuals with disease who have the marker of interest. Okay, and this is A divided by A plus C. So this is affected divided by the sum of the column. So, in other words, susceptibility, I'm sorry, sensitivity is how can you define it, in other words? It's a proportion of true positives, right? Among affected individuals. Specificity is a fraction without disease who do not have the susceptibility genotype. So this is D divided by B plus D. D divided by the sum of the second column. So this is, again, in other words, a proportion of true negatives, right? Then let's move to positive predictive value. It's a proportion of individuals with susceptibility genotype who have or will develop a particular disease. So this is A divided by the sum of the sum of counts from the upper row, A plus B. Then a relative risk ratio, another thing that I'd like you to remember, is defined as disease prevalence in carriers of the susceptibility genotype over disease prevalence in non-carriers of susceptibility genotype, which is A divided by the sum of the first row, and then over C divided by the sum of the second row. So this is relative risk ratio. We talked about sensitivity, specificity. Positive predictive value is based on the previous ones, and negative, sometimes we need negative predictive value, which is D over C plus D, which is proportion of individuals without a susceptibility genotype who do not have or will not develop a particular disease. Any questions about this part? So let's talk about genome-wide association studies, or GWAS in short. It's something that is used as the primary method to find genes for quantitative traits. So we talked briefly about schizophrenia, and I showed you a short list of candidate genes that are involved in this condition. But that also applies to many other quantitative traits, and you can hear it like in news that okay, there was a gene found for X or a gene found for Y, like a gene for homosexualism or anything. So typically the news coverage is very naive, but what this is about, or the studies that underline these findings are based on GWAS. And so what is GWAS? The goal is to find connections between a phenotype, and like I said, typically it's a quantitative phenotype like height, type 1 diabetes, and so on, that are known to be heritable, but they have very complex inheritance patterns, like multifactorial. There's some, so we need like whole genome genotypes. So the specific goals are to identify statistical connections between points or areas in the genome and the phenotype. So ideally we want to find genes or even specific regions in a gene associated with the phenotype of interest. And we want to get some insights into genetic architecture of phenotypes, like are those many small genetic effects dispersed across the genome, or are there few large effects, like some maybe a single pathway concentrated in one area, like this MHC complex, a major histocompatibility complex. And that's the ultimate goal is to build statistical models to predict phenotype from a genotype. So basically, that would be something like, show me your genome and I will tell you what disease you will get. So it's something that is more and more common in genetic tests these days. Okay, let's take a short break and we'll get back to it after the break.